Howdy everyone, you're listening to Blue Jeans and Boots, where genetics grad students talk about how to navigate a PhD program while trying to stay sane. We also invite faculty from our interdisciplinary program here at Texas A&M onto the show and pick their brains about their research, what it's like being a principal investigator, and their journey to becoming a professor here at A&M. In this episode, we'll be talking to Dr. Masako Suzuki. Dr. Suzuki is an assistant professor in the Department of Nutrition at Texas A&M University. She received her DVM at Iwate University in 1998 and her PhD at Gifu University in 2002. Her lab's research focuses on how the prenatal environment, such as vitamin A deficiency, affects the offspring cells later in life. My name is Samantha Foster, and I'm one of your hosts today.
And my name is Veronika and I'm your other host today. Thank you so much for joining our podcast today.
Thank you so much for your introduction, and then thank you so much for your invitation.
Yes, thank you. So we were curious about how did you decide what your lab is studying right now? When did you get the influence, or when did you decide that you wanted to end up in the field?
Sure, it's a very great question. Actually, my PhD is I study on the wound healing from the veterinary medicine, and at the PhD I studied how the DNA methylation affect the gene expression profile, and then I moved to the New York, and then as a postdoc I also studied the DNA methylation, and at that time I'm start thinking about how I can have my lab, my own self, and then starting what's the well known and then unknown, and then I decided to go into the nutrition as a study.
Mm-hmm. That's amazing. So you were at a different institution before coming to Texas A&M, right? Mm-hmm. So how has the past few years at Texas A&M been for you?
My experience in here is amazing because everybody is so nice, and then everybody is so sweet. I love it. I love here. Yeah,
that's good. That's good. I mean, I think we as well. That's why we joined the program to be here.
Yeah, our program is great, no?
Right?
It's really good. I love it.
Yes, it is. Did you always wanted to be a professor? Is that something that you went to grad school with this intention?
Well, when I was a kid, I want to be a veterinarian.
Mm-hmm.
And then that is why I went to the vet school. And then that's what I wanted to be. But it's kind of the retrospectively thinking, because I want to be some kind of a job always working with the dogs. And then that ends up the shifted the veterinarian. But when I entered the vet school, I found I have a cat allergy. Okay, so how can we work as in the clinical setting? And then I decided, okay, I want to go to the PhD, and then after that, I kind of want to go to the industry, but somehow I end up being a professor.
Mm-hmm.
Yeah.
What do you like most about being a professor versus what you originally thought about, going into industry?
The one thing is being in the PI is I can design what I want to do.
So the creativity and the freedom.
Yeah.
Of it. Mm-hmm.
So that's very different from well, I cannot say the perfectly because I don't know about how it works in the industry.
Mm-hmm.
But a lot of my friends are saying the industry, they need to have some sort of the plans, what they are going to do. But in the academia, we can design myself, and the things what you want to learn and what you want to study is our our decision.
Mm-hmm. So can you give us a brief introduction into what your research is about here at Texas A&M?
Sure, of course. Currently we are working on how the prenatal environment affect the disease risks later in the life. For example, if the mother do not have enough amount of the micronutrient, then it can increase the likelihood of the disease of the offspring later in life. But there is no clear mechanism has been identified yet. A lot of the people saying there could be epigenetics because this kind of engraving those the long-term memory could be triggered to becoming a disease. However, what how those kind of the epigenetic information can be triggered to the disease is not understood yet. So that is our like the major focus. And then also what so far we are finding is the nutritional status in the body. Let's say like the vitamin A status in the circulation level or the storage level are very much like the genetic variation dependent. But we don't know like what kind of the genetic variation contribute that the variation to like the micronutrient status. So we want to have to know the mechanism, how it affect, and then also like the genetic variation, how it affect, and then finally it combine the two, what's going on. So that is our study.
Mm-hmm. So those would be kind of like the long-term questions that your lab is focusing on.
Yes, it's true.
Very nice.
Yeah.
So have y'all found that the female vitamin A deficiency, like their babies are also going to be predispositioned to have deficiency as well, or does it maybe not work that way?
It's a really hard question to answer in terms of like the human setting. Let's say like if you find that someone is a vitamin A deficient, in terms of like the really like severe vitamin A deficient, they should be treated no matter what. So like we cannot do those kind of study. But a lot of like the evidence is showing as like the vitamin A deficient mother, the baby can have increased likelihood of the the symptom. So it cannot completely link because of the vitamin A deficiency is triggering the disease state. But the accumulation of the evidence could be. But the mouse study, we can treat like the vitamin A deficient and then like the showing like the how it affects. So it's not like the currently I cannot say it like the human setting.
So what does it look like when a person is vitamin A deficient? Like what kind of symptoms does the person have?
Oh, the person is not having enough vitamin A. Mm-hmm. The famous one is a vision.
Okay.
Right? Because like the night vision, like the builder, that is a vitamin A, retina, right?
Yes.
So that's the most likely, like the very well known the symptom of the severe vitamin A deficiency.
So you said you only focus from like the female standpoint, but also do you focus on the male as well? Or?
That's a great point. Then currently we are focusing on the prenatal, the environment. So it's of course like the prenatal, always like the mother, right? So that is why we are focusing on like the women, like the maternal condition. But of course, the prenatal, actually like the vitamin A the status in the pregnancy state can contribute for like the disease status later in the life. So like the next question for our study could be like the including the paternal effect as well.
Mm-hmm. That's interesting, yeah, because I feel like a lot of diseases were mostly studied from like the mother's perspective, but like I feel like with more research and more focus, there's been shifted to the paternal side as well when it comes to this kind of stuff.
Yeah, that's a great point. So like we are thinking of the parental effect. Mm-hmm. So like sometimes people saying like if we are saying like the maternal effect, some people are saying like is that because like the kids have the diseases because of the mother? No,
it's not always true.
No, so like we have both like the parents have the responsibility to get the disease or not.
Yeah.
So your research, is it primarily in mice? Are you able to do anything in vitro at all, or is it primarily like in vivo with the mice?
So currently we are working on the mice because like the prenatal effect, we cannot do it
right.
with
you can't replicate that.
In vitro, right? But probably like in the future, maybe like we can do it kind of like the generating organoids and like the testing it like the in vitro like the embryogenesis. Maybe like that could be the one of the direction. But currently we are mainly focusing on the mice.
Right. Yeah.
So let's say you had all the resources and all the money in the world. what would be like a big project or experiment that you would do?
Ooh, that's interesting question. What I want to do? You know what I want to do is I want to do like a really big the multi-omics study, because most of the time like that we are working on is like the epigenome, transcriptome, or the genome-wide association studies, or the microbiome. But if we combine everything together and then like the seeing like the phenome like, so that's my dream. But it could be cost a lot. And then also I need to have some other collaborators like they're working on that much direction. Yeah. Because it's a science. Science is always a team.
Yes.
We need to find a teammate.
Good.
Yeah.
That's a good answer. Thanks.
Yeah. So you have 2 younger grad students in your lab. What has that kind of been like, mentoring them towards, because you're mentoring 2 younger at the same time, right? So you're not having a more established grad student who's helping out. So how has that kind of been for you? How do you kind of manage that? And what is your mentorship style, I guess?
I think they're pretty mature. And then I don't feel like that they're like the young PhD student. And Mariani started her, in my lab is the earlier back to when I was at the Einstein. So like when she joined here, she's already in the 2nd year and in my lab. And then like the Brighton, she's also amazing. Like they're not like the young, like the PhD. They're almost like the very well, like the mature and then very strong, like the PhD candidate. Actually, I cannot say as a PhD candidate, right?
PhD student.
Yeah, PhD student. Before the qualifying, the prelim, I shouldn't say it as a PhD candidate, but they're really good. So I'm very happy and then very honored to have them in my lab.
Did you have previous grad students when you were at Einstein, or?
Actually, I did not have the chance to have my own PhD student in there because I started in 2020. So that means in the middle of the COVID.
Right.
And then means like I had to close my lab for about a half year. And like another half year, I have kind of like having like a occupation, like Wait, how can I say it? As like we cannot have the people in the full occupancy. Like during the COVID time, my lab cannot have a full occupancy. How can I say it in English?
I mean, I thought that was pretty good. I think that's yeah.
Okay. You can like edit like whatever I said doesn't make sense. So that's so I didn't have the chance to have a PhD student over there, but I had the chances to see the rotation students, and then also like while I was at the non-tenure track faculty, I did supervise a lot of like the PhD student over there. So I learned from them a lot. Yeah.
So since being at A&M, what do you think has been like a challenge so far as somebody who moved their labs here and has been running for like a couple years now?
So like the first challenge I thought, and then was like my lab is a lot of like the bioinformatics study, and then I also like having like mice or to generate the data, right? So we call it as like the wet season and then the dry season, and then like I moved everything at the wet season first back to Einstein, and then after all the data generated, I moved here and then doing like the dry season. So like the dry part is easier when we move because like we just need to have like the HPLC system up and like the computational, and then everything can be dry. But then when I started having like the wet lab, it had like I need to like adjust like what we need to do, and like I got the protocol done, and then like the environmental health check done, and then like the lab-wide training done. Of course, every institute have like the different regulations, so like I need to fit to that one. So that wasn't challenging, but other than that, it was amazing. I like it. Yeah.
Good.
Yeah.
What do you think has been the most rewarding moment so far?
The rewarding moment. Someone came to my office and then saying like there's something exciting, the data. And then the person is super excited about it. That is a reward moment. If you like just like come like the result without like excitement, it's so sad. But if someone is like coming like look, I have this, and then that's me like, yay!
So you celebrating with your students?
Yeah, of course. Why not,
sweet.
right?
Yeah, of
Because
course.
like you got it, something like the showing us something interesting, like the why not, like they celebrate,
Yeah. It is always the best feeling when you have good
right?
data.
You know, like that is why we're doing the science, right? The science is fun always.
Absolutely.
Yeah.
Do you think there is something you wish you would have known before having a lab?
Good question. So like I think my career is almost like more than 10 year later than the typical, like the academia, like, like the career. So I didn't know when is ready to be in the PI. So that was kind of the big struggle always the time. Always I think I'm not good enough. Always I'm not good enough. And then like some point, I need to moving forward. But I kind of hesitate to step forward. That was kind of the struggle. And then when I got my first R1, my mentor, Dr. John Greedy, told me, oh, Masako, you are the only one who can protect your lab and who can protect your lab member. You have to be there. You have to do something. And that is make me okay, step forward. Mm-hmm. But still I'm feeling yeah, I'm not good enough. That is
that's imposter syndrome,
right? I think I'm the typical one. Yeah.
Yeah. I think it's really common in academia for sure. But how have you kind of worked through that, I guess? Would you give any advice to maybe some grad students who might be struggling with imposter syndrome?
One thing I can say is you guys are great. That's that's the one maybe somebody when I grew up in the Asian country, the women always been the supportive side. And then that makes me always I'm not good enough. But when if I can see my when I was 20 or something, and then maybe I will say you're great, and then that's kind of they keep saying you're great, you're amazing. That is the helping to the girls to realize how great you are. You guys are great. That's for sure.
Oh, thank you.
That's very sweet.
So I guess kind of along the same lines, is there anything that you do outside of the lab that kind of helps you disconnect or relax, or just that you enjoy doing?
Outside of the lab, I have 2 dogs. They're my the buddies.
Yeah.
And then one is from when we were in New York. She joined us, and she was the 2 or 3-year-old, and then she's about 18 pound, the small dog. Mm-hmm. And then after we moved here, we have more space, and then we decided, probably we want to have another one, and then we are looking for a small size dog, and then I found the one of the rescue. And then saying the another one is coming from rescue, and then we are looking for the rescue, and they're oh, this is cute, and then she's a puppy, and then I called the organizer and then saying yeah, she's a small puppy, okay, and then she's saying they're saying yeah, she could be about currently she's about 14 pound at the 5 month, but probably she will be about 30 max 35 pound. And then we went there, and the gigantic puppy is a big smile. And we were wondering, what? Is the small puppy, and then the host mother said, yes, she's a small puppy, and then
So she's gonna be huge.
Basically.
yeah. Currently, she is about 32 pounds at the 7 month.
Oh, wow.
She's still a puppy.
Yeah, she's still a puppy, and she did not recognize how big she is. And another one is we call Coco, and then Coco-chan is always being calm on my lap, and she's tiny. And the Lupin, the another one, is she thought she can do the same thing.
She can.
Oh yeah, and then we call her as a Texas-sized small dog.
Mm-hmm.
Yeah.
Yep. Yeah.
Everything's bigger in Texas.
Yes,
Yep.
that's so true. Well, she's sweet. She's
Yeah.
really the same. The college station people, everybody's sweet, right?
Yeah.
Same.
Did the two dogs, even though they're different sizes, do they get along and play really well? Or
The first the few days, the old dog didn't like her at all. So that was a bit she's not good at playing with the other dogs. She preferred to be independent. But but I thought maybe she's good at being independent, but she will kind of have the dog friends. But the puppy is always a puppy, right? They wanna she wants to play, and then hey, I wanna play, and then Coco-chan is kind of hated. She looked always like this.
That's sweet. So how would you say you balance the demands of research, teaching, mentoring, and having your own free time as well? What strategies do you use that kind of work for you effectively?
That's hard to answer because it depends on the time of the year, right? So sometimes I'm very much focusing on my research. Sometimes I need to focusing on my grants, the teaching. So time by time, modifying it. And then,
so it depends on the time of the year.
Yeah. Sometimes I need to have the time with my dogs to get relaxed. Sometimes I just want to focusing on to do the analysis, and then before the writing the grants or the writing the manuscript, I need to focus on. And then for the teaching, I need to be prepared. So
do you teach classes here at A&M?
Yeah, sure.
You do?
Yeah.
What do you teach?
So the last semester, last 2024, the fall, that one that I teach the nutrition through life, which is my favorite one. It's for the sophomores and the junior people, and teaching how the nutrition stays through the life. That was a fun class. I liked it.
That sounds nice.
Sounds interesting. Yeah.
And then another one is I teach the scientific communication. The student write the it's little the publications and the report and do the public speaking. That was really fun because I can learn a lot of things from them too.
Yeah. So what do you like most about teaching?
I like to seeing a lot of the student talking about the different things. I can learn many things from them too. That is the best part.
Yeah.
So the diversity of people's interests and ideas.
Yeah, that's just fun being the university, right? Everybody's different. That's just the color. That's the beauty.
Do you only teach undergraduate classes or any graduate classes as well?
Not yet for the graduate courses, except at the seminar, no graduate course yet. But hope in the future, yes.
Yeah. So you did touch up on the seminars. So just a brief introduction. Our program has seminar series, and every Monday we have a speaker that's either from Texas A&M or an outside speaker. And so you have been involved in the GENE program a lot, right? So you've also been in charge or the faculty advisor for the seminar series. And you were also recently awarded the Student-Elect Faculty of the Year Award.
Thank you.
So I wanted to ask, how has your personal experience been in the genetics program? Because you are very involved, right?
I really like this program a lot. The A is just very diverse, and then I can learn from like the many things. For example, at the seminar, it can be starting from like the Drosophila to the plants, and then like sometimes like the population genetics. It's very different, and then like every time I can learn from the basic to the translational and a different field, and then that is really good to see what's going on the genetic field. I like it a lot.
What do you like about, because you're head of the seminar committee, so what do you like about being involved in the seminars from that direction?
So like the seminar is the student driven. So like I'm being just like the supporting, helping from the background. I'm not doing anything. Almost like the students are doing a wonderful job. I'm just like the cheering up. Great job, great job, great job. That is my turn.
That's more than enough, honestly, sometimes. Yeah. So going back to how you organize your lab and I guess like mentoring your students, how would a typical week look like for you?
So during the semester, I always have a one-on-one meeting once a week, starting from the new undergraduate and like the PhD student, and then the staff members. So almost like the stretching out a lot of like the time, but I really like it because like the lab members are my family, and then the team. I want to know if there anything that they need help or not. Mm-hmm. So that's the time I'm spending a lot of time to make sure everybody's okay, everybody's happy.
Yeah, that's awesome. Yeah. What advice would you have for current grad students or incoming grad students? Or if you have different advice for the different stages, or...
Wow, that's a tough question. No, it's a very different of the career stage, right? Mm-hmm. So
yeah.
For the current PhD student, enjoy your life. No, mm-hmm. You are doing fun stuff. You should be happy, and then that's the thing. I believe. And then for the incoming, the student, the first thing is have a wide vision. Mm-hmm. There are many options available. Don't stick into the one. Probably sticking the one is great, but before you decide, see many options as many as possible to find out what is the best option for you.
Yeah, I think that's something that we've been told as well when we were doing our interviews, just trying not to focus on one research area, but having an open mind and going into rotations, trying out maybe science that we didn't even consider before.
Yeah.
And I think people usually do forget about that there's so many diverse topics that you can research and keeping an open mind.
I think it's something our department is really good about, is encouraging you to, during using a rotation, to explore something completely different than what you normally would, and just getting a feel for just everything, because you could be surprised.
I know.
You never know.
That's I think that is the one great thing in the umbrella program, right? Because you can see many options, and if you're going to the admission to the one lab and stick to the one lab, maybe you are going to miss the opportunity to see others. But the program, we have a lot of the background, we have the diversity, so you can do a lot of things to try and then to find out if you like it, don't like it, so like it, very much like it.
Yeah.
So that's the option.
Yeah. What kind of advice would you give to people who want to have a career in academia?
Believe yourself. You can do it. Mm-hmm. So don't hesitate to step forward.
Yeah. And do you think that people should be prepared differently during their graduate studies if they want to go to academia compared to go to industry?
I never go to the industry, so I cannot answer the question. But the being prepared, the different. No, I don't think so. The important thing is you like what you're doing or not.
Absolutely.
Yeah. So the members in your lab, are they more interested in going into academia or into industry? What are they kind of leaning towards?
I believe a lot of people want to go to the industry these days, right?
Yeah.
How about you? You want to go to the industry or you want to go
industry?
Yeah. How about you?
Me too.
Right?
Yeah.
So now it's a 100% they want to go to the industry. So that is a trend, right?
Yeah. I feel like I don't think I've considered much going into academia, even when I did start grad school and people were saying like, oh, you never know, you might decide on your way. So I've been kind of like, okay, maybe I'm not going to set my mind on one thing, but I don't know. I feel like the industry field has just become a bit more attractive than academia.
Yeah. And I'm very much someone who I like to be hands-on and I like to be doing the experiments day to day. And I don't think I would like being in more of an administrative role, like more consistently. Yeah. So that's a big thing for me. Yeah.
Well, in the academia, also there's the many the opportunities too, the being, let's say, the research scientists. They're very much the focusing on the expertise on their own the project, right? So it is not the academia being the PI, but the academia in let's say if you're really good at with on the techniques, then the facility job will be very much hands-on, and they're the foundation for us doing the good science. That is another the possibility for your career, the career path as well.
Yeah.
Absolutely. What do you think has been the hardest part about being a PI? What do you think has been the hardest part about being a PI?
Hardest part. Let me think. Sometimes I feel the bit lonely. When I was the postdoc, when I was the non-tenure track faculty, and I always have someone to ask to get the guidance, let's say the direction of the guidance. But as a PI, I have a responsibility to decide, and then sometimes thinking myself a lot, but sometimes I really need to seeking the guidance. But sometimes I'm feeling I might need to be the independent. I shouldn't asking all the time get the guidance. So that makes me the different from what I was in the before.
Does your lab collaborate with other labs by any chance for projects?
Yeah, in my lab I have a lot of collaborations in the A&M as well as the Albert Einstein, then Columbia University, and then University of Virginia, UVA.
That's amazing. Okay, wow. That's a lot of collaborations.
Yeah.
How do you manage all those different collaborations?
Well, not managing it. What we like to do is what is supposed to go. So we have the discussions and find a way, and then that's how we are going to go in the direction or not. We have the discuss, and then the deciding what is going to be the next, and then the deciding how are we going to the submit a grant. So that is not the managing, but the time wise, sometimes it's a kind of the tough time. Of course, the on time and off time. So the one that the two projects on ongoing and on time will be a bit hard to find out a great time. The scheduling the time, but the most of the time we can find a way.
Right.
So do you work with all of them at the same time, or is it does it depend?
I prefer to work as the multi-project at once. So I think it's my characteristic is I'm not good at focusing on one thing too long time. I need to have a switching my brain. So one time I work this, and then, okay, after the lunch, I need to switch. After the dinner, I need to switch. Something like that.
So you just need to be like recycling through it to keep it like going, take a little break from it, come back to it.
And then, one day we are analyzing data, and they're like, oh, wait a minute, I think I can apply this one to the other project as well. So kind of like the thinking a lot, but that's like the better way to doing like the project at once.
It's good.
Yeah. Can't handle multitasking as well as it seems like you can, or I would have multiple things going on at a time if I could, but I guess my brain doesn't work that way.
So are there multiple projects in your lab just happening at the same time? Multiple like different focuses, I guess, regarding like vitamin A deficiency?
Yeah, currently we're working on the vitamin A deficiency and then genetic variation of the vitamin A, and the Brighton is working on the vitamin A, the status, like the prediction model. And then the UVA, I'm working with the preterm birth effect, how it affects the kidney, the diseases later in the life. And then on the Columbia University is the vitamin A deficiency in the lung. And then Fordham University, I'm working on the brain multiome study. A lot. Yeah. But it's fun.
Absolutely, of course.
Yeah.
What has been, in your opinion, the biggest finding by your lab?
Oh, the what I kind of surprised is even if we are eating actually we cannot say it one of we fed exactly the same diet, and they're giving the same environment, vitamin A status is pretty much the genetic variation dependent. And then a lot of people are saying because it's mostly doing a human setting, right? And then the human we cannot do it because the vitamin A is essential micronutrient, depends on what they eat, what kind of the beta carotene or the lectin ester level. So they have the different. But the one that we are doing the animal, they can do the same, they can have it. But the most the fascinating the thing is their difference is 2 times, 3 times different between the high strain to the low strain. If we consider that is the hypervitaminosis or the hypovitaminosis in the human, right? It cannot be the 2 times, 3 times. But in the mice, what we found, 2 times, 3 times difference. And then they're healthy. What the heck is going
there? Such a big difference. They're healthy?
Yeah.
Yeah.
They're healthy. They're just the different mouse strains. One is the wild derived, and another one is coming from the lab strains, and they have the 2 times, 3 times difference, and they're healthy. They're okay.
You're using the same diet for both of them.
Yeah,
but just the genetic variation is what is making such a difference.
Yes,
that's interesting,
right? That's if you think about it, you're the vitamin A status is 2 times higher than me. Right? It could be something.
Yeah.
Not in the mice. So that is my the fascinating finding after I moved to here.
Yeah.
But in humans, it would be if you did the same diet, the genetic variation wouldn't, I guess, affect it that much as in mice.
We cannot test, right? Who wanna be eating the same thing entire life? And the testing it at the it's an ethical issue, right? But if they are eating exactly the same diet and at the age 20 and then testing it vitamin A level, probably, but who knows? We cannot test that.
Yeah.
So how much variation do we see in humans as far as vitamin A levels?
In terms of the difference means the ranges?
Yeah.
Not 2 times, 3 times.
Right.
Yeah.
Right.
Sorry, I don't have any the clear answer. Yeah.
Yeah. It's just interesting.
Yeah.
So what do you think could be potentially a project, maybe not done in your lab, but an ethically acceptable project regarding vitamin A deficiency?
Well, again, those like the deficiency, the project. My personal feeling is we shouldn't have that one.
Mm-hmm.
Because it's ethical,
Mm-hmm.
everybody needs to have enough amount of the vitamin. Being healthy. Everybody needs to be healthy.
Of course.
So, and we cannot do that type of study in the human setting.
Mm-hmm. Sometimes,
The people are doing with the vitamin A deficiency high prevalence area. That's cool. Some area have a higher rate of the vitamin A deficiency, and then they tested with that regional level the variations, but I don't think it's the great idea.
Yeah. So maybe comparing different diets, because different diets would have different levels maybe of vitamins.
Yeah, that'll be the great one. But the issue is the retention rate of the eating the same diet is not so great.
Mm-hmm.
You wanna eat the same stuff the 3 months?
No,
right? Let's say you wanna Mediterranean diet is great. You wanna eat a Mediterranean diet every day on the the similar stuff on the year? Maybe not. Sometimes you wanna have something else. Like the cheeseburgers could be great, right?
Yeah. Could you look at different regions of the world where overall the diet is a little bit different? Of say the United States is kind of, I guess American not great food. And then if you look at other areas of the world that have slightly different kind of.
Yeah, there are many different in terms of cuisine is very different, or the cooking style is also different, but also genetic variations are also different, right? The Asian countries have the different type of a cooking style, but the Asians and then compared to the Hispanic populations, we have the different the genetic background too. So we cannot simply compare A to the B.
Yeah.
It seems like there's a lot to be still figured out, I guess.
Yeah.
And it's hard to figure out because it is a vitamin deficiency field, so
yeah. I think in the future is what we want to have is the precision nutrition.
Mm-hmm.
So let's say once we have all the mouse data accumulated and then everything this genetic variation is contributing to the nutrition status of the A nutrition, then we can accumulate all the the information, and then that is going map it back to the human. And then if the human setting is also showing the same trend, then we can have, you have this and that genetic background, and then your the dietary style of this, you might have more some sort of let's say you might have a little bit more the vitamin A compared to the other folks. Those kind of the precision nutrition is going to improving our health. Mm-hmm. That is our the future goal.
So that's the overall idea that we're working towards.
Yeah. Mm-hmm.
So how would you translate some of this mouse genetic variation into understanding the genetic variation for humans and how that would affect it?
So that is also the very important question, and then there's no a single method to do it. And for example, we have we identify the genetic variation in the certain gene, but if it's in the let's say if it's in the coding region, then the changing the protein function, that's easy. But if we identify the genetic variation in the promoter region or the enhancer region, and then we don't know what is the effect, and then where it kind of on the comparable the region to the human. So that is could be a little challenging. So we'll see.
Yeah. Well, thank you so much for coming to this podcast. I think we learned a lot about a field that we were not familiar with before.
Thank you so much for having me.
Thank you. Today's episode was hosted by Veronika and Sam Foster and produced by Anthony Matarazzo and Charles Mitchell. This podcast is brought to you by the Texas A&M University Interdisciplinary Graduate Program in Genetics and Genomics. If you have any questions about our program here at A&M, please feel free to email us at bluejeansandboots@gmail.com. You can also find us on Facebook, Instagram, YouTube, and X at Genetics Podcast. Thank you for listening, and we hope you tune in next time.